All right, everyone. Welcome to today's Bioinformatics for Precision Oncology session. Today, we're going to be discussing data sources and databases that are commonly used in oncology research projects and that you can leverage in your projects as well. This portion of the program is designed for you in order to prepare for the independent analysis steps. So please think about the types of data that you'd like to use, specific projects you might be interested to work on, and how these steps that you've been learning will help you to find the right type of data for a project of your own. Let's briefly go over course progress. So this is the graphic since last time we met on the 26th, and these are some of our leaders, and you can see that some of you spent quite a bit of time on the server as well as on the education side. Let's go through a couple of quizzes regarding the material that we talked about last time. I have a couple of questions. Most of these are related to the PDX breast cancer model. So I hope that you had some time to review that information over the weekend. So for this one, it says, which of the following mouse types are used in the PDX breast cancer model that have severe combined immunodeficiency, but also have normal natural killer cells, macrophages, and granulocytes? And as always, you can post your responses in the chat, and I'll be reading those off. So Mikhail says 2 and 3. Zenab says 2. We'll also be going over these a little bit more today, just kind of as the review part. So don't worry, but the correct answer is going to be 1. SID-CB17. What I can say about SID-CB17 is that these have severe combined immunodeficiency, but they also have normal natural killer cells, macrophages, and granulocytes. Since we got some answers for 2 and 3, I'll read off those. SID-NOD have severe combined immunodeficiency, and they don't have mature T cells or B cells. And then when it comes to athymic nude, these don't have a thymus, and so they're unable to produce T cells. So here's a photo of our mice. We'll be also going over this again here just a little bit on some other slides. So hopefully we can drive these points home before moving on. Let's try another one. This one says, which of the following molecules are likely to interact with cell receptors on the ovary, uterus, and breast? I think that should say in, not on, but I was writing these questions this morning as I was preparing for our lesson today. So Zenab says 4. Sumi says 4 as well. And so if I tell you that the correct answer indeed is 4, can any of you tell me what the common precursor molecule for these molecules is? So I guess my question is, all of these molecules are derivatives of what molecule? And they all kind of closely resemble it. They have a 4-ring structure. They're all aliphatic molecules. They have a polar head and the rest being hydrocarbon. So all of these molecules are derivatives of cholesterol, as are all of the testosterones. steroid hormones like testosterone. Let's try another one. So each of these molecules passes through the bloodstream and binds to cells that possess a receptor for it. Which of the following is an accurate descriptor for what occurs upon receptor binding? So Sumi says that both 1 and 2 are correct answers. Does anyone else have any guesses? Zenab says that only one of these are correct. So in this case, Sumi is going to be correct. The correct answer is going to be 3. And so that makes that, so these intermediates function to traverse their target cell's plasma membrane via a process of simple diffusion and onto the nucleus where they bind to the estrogen receptor. And then the other statement that's true is that these intermits bind to hormone response elements and activate the transcription of target genes. Can any of you tell me the difference between active transport and simple diffusion, and why these molecules may be perfect for simple diffusion? So Sumi says energy required. Yes, so for simple diffusion, there's no energy required, and active transport requires the input of cellular energy. These molecules, since they're cholesterol derivatives, are able to pass through the membrane because they are in large part very similar to the lipid structure of the membrane itself. And then once these estrogens traverse the cell membrane, they go onto the nucleus, and then they are going to bind to hormone response elements. And then those hormone response elements are going to ramp up the transcription, translation, and then inevitably the translation of target genes. So let's try one more. It says, which of the following methods allows us to find pathways or gene sets that are enriched with up or down regulated genes of interest? The answer choices are gauge, any pipeline that includes PCR clean and trim-o-matic, any pipeline that includes FASTA files, or a pipeline that includes FASTA files and then includes gauge. I don't think any of you are fooled. The correct answer here is going to be gauge. So here's a photo of one of the outputs from gauge. Remember that, and we're also going to talk about this a little bit more today, but remember that with these, we have our genes that are either upregulated or downregulated, and this is going to be indicated here on the top right, where green is going to be downregulated and red is going to be upregulated. And so we're going to be talking about these things a little bit today before we move on. So we can go ahead and start a little bit with that review. So remember that in order to study the biology of cancer and understand it, we focused on our PDX models. We had 4 different cell types: SID-NOD, SID-CB17, athymic nude, and XID. Remember that XID mice don't have a thymus. And what this means is that they don't have functional T cells. These have mutated B cells. Athymic nude mice don't have a thymus, and so they don't have the ability to produce T cells, but they have normal B lymphocytes. And then CB17 was the one that we talked about in our quiz question. This one has severe combined immunodeficiency, but they're able to produce normal natural killer cells, macrophages, and granulocytes. And then SID-NOD mice have severe combined immunodeficiency, and they are unable to produce T cells or B cells. Remember that in the project, we also looked at the different types of tumors in our data. Triple negative breast cancer is any type of breast cancer that does not have express the genes for estrogen receptor, progesterone receptor, or HER2. And then remember that HER2 stands for human epidermal growth factor receptor 2. We also talked about biological interpretation of our differentially expressed genes. And we found some intriguing insights into the underlying differences between ER positive and TN subtypes of breast cancer. In our data set, we found 20 genes that were upregulated in TN. And then we saw that 4 of those belong to a class of molecules called non-coding RNAs, which are ncRNAs for short. And that specifically those non-coding RNAs belong to a subclass called long non-coding RNAs. And then we also found that there were 2 key factors in neutrophil migration that were differentially expressed in TN breast cancer. These were TNF alpha IP6 and CXCL8. TNF alpha here is going to be a player in neutrophil migration, and then CXCL8 was a ligand for CXCR2. And so because these were downregulated, the ability to induce chemotaxis of neutrophils as well as the priming of neutrophils was attenuated. We talked about gene annotation and pathways. And so here's I guess an example that we used here using the annotation pathway. And in order to do this, we found different information, and this included decorin. And we also talked about how decorin is a protein that is involved in the regulation of the cell cycle. And so it's also involved in the regulation of autophagy, endothelial cell and angiogenesis inhibition. And this process is mediated by a high affinity interaction with VEGFR2. VEGFR2 is short for vascular endothelial growth factor receptor. And this leads to increased levels of tumor suppressor gene that's called PEG3. Other angiogenic growth factors that decorin inhibits are angiopoietin, HGF, which is short for hepatocyte growth factor, and then PDGF, which is short for platelet-derived growth factor. And I see that Sumi has a question regarding the annotation pipeline. Towards the end of the session, we can take some time to go onto the server. If you don't mind, I'm going to get through the material that I already have, just so that way questions on forum. And what this allows me to do is to kind of know which concepts you're struggling with, and then oftentimes what I can do between sessions is try to design some content around that area so that I can make sure that I have everything kind of prepared. Pipelines often take a little bit of time, and so to do that on the fly is kind of difficult. But perhaps Mohit and I can, at the end of the pathway, or at the end of our presentation, kind of discuss this with you. But if you can, post on the forum, and I'll do my best to have that answered to you at least by the next time that we meet. Remember that this is a differential gene expression analysis that we're kind of doing here, where we go through our pre-processing steps, which were PCR clean and trim-o-matic. We use mapping on transcripts with bowtie, and then we can get our expression levels. And then DSEC2 is an algorithm that's been designed for differential expression analysis. Another one that you should probably look into is EdgeR. There are differences between those, and we actually teach courses on this. So if you're interested in that, I can probably pass that information along as well. But let's go ahead and move on and talk about the gene set pathway enrichment analysis. And so remember that the results of GAGE is something that looks like this, where you can see inside a pathway. And for example, this is the regulation of the actin cytoskeleton. On the right side here, you have the B cell receptor pathway. And so last time I think we talked about how you can see many that are going to be either up or down regulated. So this is kind of cool because if you can go in and kind of look at a pathway and find multiple instances of something that may be interacting, and then kind of look at a pathway as a whole, and it's really cool that we're able to kind of do this in an automated fashion. A lot of the material that we're going to be talking about today can be found in Transcriptomics 3, which allows for more advanced analysis of gene expression data. And so here you'll be exploring some different methods for identifying groups of samples without prior knowledge. Remember that this identification of groups without prior knowledge is called clustering. And this is a unsupervised machine learning method. And then you'll also be in Transcriptomics 3 examining methods for developing classifiers from known samples. And this is the allows you to classify unknown samples. So the sections of this course are, first you're going to learn a little bit about the associated data set, and then work on data preparation. And then there's a review of principal component analysis, or PCA for short. The second section second section is going to be over unsupervised analysis and clustering. And you'll learn about hierarchical clustering, which we're going to talk about today, and then k-means clustering. There's also classification, which is unsupervised analysis, and this is the topic of section 3. Here you'll learn about classification, feature selection, the LDA, or linear discriminant analysis pipeline, and then there's the stepwise linear discriminant analysis pipeline. There's also the SVM pipeline, and then we'll be learning how to do biological annotation of 17 informative genes before concluding this course. Machine learning is the scientific study of algorithms as well as statistical models that computer systems use to perform specific tasks without using explicit instructions. This technique relies on patterns and inference instead, and it's seen as a subset of artificial intelligence. But how does this fit in with the actual biological steps that we're doing? And then how do we, for instance, plug all this into the computer? Well, there are certain steps to this analysis that we can go through, and I will talk to you a little bit about these in order. So the first step of this is going to be the biopsy process. And in step 2, this is going to be separated into tumor and non-tumor cells. And here, these single cell suspensions are going to be magnetically labeled using the non-tumor cell depletion cocktail, and this is contained in your tumor cell isolation kit. So you'll follow these steps according to the protocol that's included with the kit. These cocktails label non-tumor cells, and thus enable the isolation of unlabeled tumor cells. DNA extraction for the tumor occurs during step 3. And in step 4, we have our sequencing and analysis. We combine our data with existing samples in step 5. And then finally, in step 6, we're going to be analyzing our results by combining the data from a given sample with other data that we already know about. By comparing them to data that we already know, we can find similarities to one of the known groups. And so for here, we're looking for similarities. And so this kind of describes how we go from the biopsy process all the way through to the analysis process. So that brings us to our session today. And now we can go ahead and kind of talk about big data challenges as well as those opportunities. So we can also discuss how the growth of high throughput sequencing, as well as how this applies to cancer genomes and transcriptomes, has produced a big data problem. And this problem precludes many cancer biologists and oncologists from gleaning knowledge from the data regarding the nature of different malignant processes, as well as the relationship between tumor genomic profiles and the treatment response. And so this is kind of why we're going to be learning about how these different data types work, and then how to efficiently process and analyze such data. We're going to be using an example from a publication that was written by Damon. The publication's titled Modeling Precision Treatment of Breast Cancer, and it was featured in Genome Biology in 2013. We here at edu.t-bio.info find this publication interesting because the authors assembled a data set of about 50 different breast cancer cell lines, and these represent the whole spectrum of breast cancer variations. These cell lines have genomic, transcriptomic, and proteomic data, and they also include an efficacy measurement of commonly used and experimental cancer drugs. So if you want to check out these projects or other projects that we have, I'm going to go ahead and post this link in the chat for you to take a look at. And there are many different projects that our company does. And so I encourage you to take a look at those if you're wanting to find more things to play with. Let's see what the benefits of RNA sequencing are, as well as what RNA expression can offer for molecular classification of breast cancer. Remember that histological analysis of a tumor can be illustrated by looking at a mammogram, and a mammogram is an X-ray picture of the breast tissue that doctors use to look for the early signs of breast cancer in suspected lumps. In this example, you can see our highlighted tumor, and then once a tumor is found, it's critical to establish whether it's malignant or benign. And then additionally, we need to find out what type of tissue that it's originating from. So once the tumor is characterized by its phenotype, we can take additional information which can be obtained for checking for the presence of specific proteins that are characteristic of cancer. If the proteins are known, then we can use a process of immunohistochemistry. And then immunohistochemistry can be used to find if they're expressed or not. So on the right here, we have our PAM50 panel. And here you can see that this is a panel of 50 genes that shows their expression. Unlike the gauge example that I gave you, this shows high expression in green and low expression in red. Remember that a PAM50 panel uses a 50 gene signature that can be tested using qRT-PCR, and that there's other types of panels. These other types of panels can be Oncotype DX, and this has a 21 gene recurrence score. And there's another one called MammaPrint that has a 70 gene signature. And this MammaPrint has been approved by the Federal Drug Administration for use in the United States. PAM50 is also now called the Prosigna breast cancer model. Breast cancer is a heterogeneous disease, both genetically as well as clinically. And so there's a need for histological, molecular, and functional classification systems that can aid in devising an appropriate treatment, as well as predicting clinical behavior. Histological classification has been considered as a valuable tool for classification of breast cancer subtypes for decades. And this has diagnostic, but kind of limited prognostic value. It characterizes different types based on their histological features, such as their glandular formation, nuclear pleomorphism, and mitotic rate. So there's been different molecular subtypes that have been identified, and these are luminal A, luminal B, basal-like, and normal breast-like. There's also Claudin low and HER2 plus. And so these have all come about via the evolution of the molecular classification system. And this has great potential for predicting therapeutic responses. Additionally, there's an improved ability to predict overall survival as well as disease-free survival. And so therefore, there's been kind of this progression of prognostic predictions that can be made. One question we can ask, though, is how can molecular subtyping be used? Because many patients characterized with such methods were observed for such a long period of time, we're able to actually now assess risk factors for the recurrence as well as survival. Over here, we see a Mayer-Kaplan curve of survival or recurrence based on 2 clinical gene panels. This is using PAM50 and the Oncotype DX that we talked about earlier. These panels separate patients into 3 groups, which are high, medium, and low risk. And based on these assessments, a treatment course is going to be selected. Traditional treatments for cancer are going to be surgery, radiotherapy, and chemotherapy. Each one of these different types of therapies can have harmful side effects, and it's known to be effective in specific situations, but however they consider that, or you have to consider that a lot of times that, you know, the treatment may be worse than what was going on before. However, since the 1990s, a new type of treatment started emerging in clinical use, and this is called targeted cell therapy or targeted cancer therapy. There's different types of treatments that have been brought about because of this. One of these is Herceptin. And what this is a monoclonal antibody that blocks HER2 receptors, which blocks it from receiving the growth factor and therefore prevents cell growth. Let's talk a little bit further about the Modeling Precision of Treatment in Breast Cancer paper. Here we see the abstract of the paper, as well as the graphical abstract, which summarizes what the author were trying to do in the analysis that they performed. As you go through this, you can see some of the yellow words that were highlighted, and these are topics that this publication falls under. Orange highlights the methods that were used, and many of them are going to be discussed in details throughout the course. From a quick reading of the abstract, we find that the authors used machine learning algorithms to identify molecular features that are associated with different breast cancer groups and associated with those in terms of treatment efficacy. On NCBI, you can find that data is organized into bioprojects, and these contain links to publications, various metadata about samples, and if there's raw data available, it can be downloaded. So here, this is how you're going to find data sets and download information about your samples. And it's going to be important for us to understand what we're dealing with and for how to do this process. So what cell line is here and what other information is known about each sample can be ascertained using NCBI. I'm going to post a link in the chat for you to NCBI so that if you're interested in finding more information about bioprojects, you can. Data is stored in a compressed format or sequence read archive. It's also called SRA for short. And so this SRA archive is going to contain runs or samples, and these are going to start with SRR, which is short for sequence read runs. So if you download and extract the SRA archive, you'll find that they contain FASTQ files, and the extensions for those are going to be _1 and _2 at the end. Looking at the metadata given with the given project, you can see that the data was produced using pair-end sequencing. This technique uses simultaneous reading for each fragment from both ends. And what this does is it improves the quality of the sequencing. In addition to the file name and its download location, your metadata is also going to show you cell line names that are used in each run and the clinical subtype that it's associated with that sample. Once you download your NCBI SRA table, you'll see that the names of each sample or run are named with each associated cell line and its clinical subtype. And these are going to be luminal, basal, cloud and low, as well as normal-like. These molecular subtypes have different prognosis, as well as morphology and patterns of gene expression. And to see whether one of the subgroups can be found in our RNA-seq data, you have to convert the SRA read files into structured data. So on the platform, you can convert the read data from SRA files into a table of expression, which will be able to analyze using various machine learning and data mining techniques. Depending on the pipeline of your choice, you can download a smaller set of 11 samples with only 2 subtypes, or you can download the full set of 52 cell lines. Our data is going to be in FPKM format. And then remember that FPKM stands for fragments per kilobase of transcript per million mapped reads. What this means is that the gene expression level is already adjusted for the total number of reads in each sample, as well as the length of the given gene. This measure is typical for RNA-seq outputs from pair-end sequencing. And so the file extension that you're going to be downloading is going to be expression _ genes _ cell _ lines.txt. So I'm going to post that in the chat, simply that so that you recognize it when it's time to download that from your pipeline. Once you download that file, you can open it in Excel and then see the contents for that. The results of the pipeline will give you your table of expression. And not only is this table normalized for read count and gene length, other transformations have already been done to the table. And these have been done in order to remove genes with normal expression across all samples. With 0 expression across all samples. And so once you eliminate genes with 0 expression, you're going to end up with approximately 17,000 out of the original 69,000 genes. RNA-seq data has been shown to have skewed distributions, unequal variances, and extreme values. Most statistical tests, including the ones that we use in our analysis, have an assumption of normality. So those skewed distributions, unequal variances, and extreme values violate those assumptions of normality. And there are a number of normalization approaches that you can use as a standard approach that would include conversion of values to log scale and adjusting the distribution to account for technical variability of your lower expressed genes and aligning the mean of distribution between all samples in your set. So on the tBioInfo platform, you can perform both of these using the quantile normalization utility. And this utility can be found on the areas of analysis page. And so as you go through, it's going to be down on the bottom right there. And then you can find the utilities and quantile normalization. Quantile normalization and other normalization procedures are important to transport distributions between and amongst different samples to have the same distribution. What this does is it removes unwanted technical variation that could cause perceived differences between your samples when there are in fact no differences in terms of the biology. For example, this could be changes in your experimental conditions. Quantile normalization is a technique of mapping original values to the corresponding quantile of a standard distribution. And here, an estimation of sequencing depth of an experiment is determined by upper quartile of its contents. Comparing quantile values instead of the raw values will eliminate differences solely rooted in technical variation, such as the amount of RNA that you sequenced, which type of sequencing machine you used, or even unexplained differences that arise between technicians that are following the same protocol. So in addition, while normalizing data, we will use, in addition, while normalizing data, we're going to be using log transformation, and this is going to make it easier to interpret differences in gene expression. So now you can use normalized data with clinical annotations that we have from the SRA run table to perform our analysis. In our next lesson, we're going to be using this data to perform clustering and then identify groups of our samples. So you can see that pretty much all we've done so far today is just to get to right here. Next, we're going to be going over clustering, which is unsupervised analysis. And then I think next time we'll be talking more about supervised analysis. If you don't mind, I'm going to pause real quick and grab a drink of water. If you have any questions, please take time to post those in the chat, and Mohit and I can help to answer those for you. All right, so let's go ahead and move on to unsupervised machine learning. Remember that with unsupervised machine learning methods, we start with a data set, and typically this is a table of elements, such as these shapes. We can use a clustering method, for example, hierarchical clustering, which is a method that seeks to identify groups based on their similarities. We can also use a dimensionality reduction method, such as PCA, that will use data variance to assign samples to groups of elements that are present. In some cases, we can use these results to draw other conclusions about our data within such a structure. And in this case, the groups are based on the shape of elements within a group. So this is the new information that this technique generates, is these different types of groups. Let's talk a little bit more about what clustering is. PCA does not identify groups or their boundaries, and so this is why we need to turn to clustering. There's many different types of clustering, and hierarchical is one of the most popular approaches. The reason why it's mostly popular is because it allows for clustering on different levels. So let's say that we have our samples organized in some space. So you can imagine a PCA 2D plot where the samples are positioned in some perspective. They're going to be positioned in some perspective that best represents the data variability. And what the algorithm does is it finds the two closest objects in this projection and then links them together. And so this is what we're going to be calling a cluster. And then we would find the next group of two, where any two linked objects are considered as a single object, and their location is going to be the center of that cluster. What the algorithm will do is it's going to run until all the objects are assigned to a cluster with all the objects in it. If you're interested in running a clustering algorithm on the tBioInfo platform, go to the data mining section and you'll find this under the unsupervised analysis section. So you'll need to upload your table of expression that you generated using the cell line data. So you can go back up to the different demo projects that we have and do the cell line demo. Let's talk a little bit more about hierarchical clustering. This is an example of something that's called agglomerative or bottom-up clustering. And it's a method that starts with attributing each sample to a separate cluster. It then finds a pair of the most similar samples and unites them into one cluster. This procedure is repeated until coming to one cluster that contains all of our samples, as we did in our last graphic. And the pipeline will take the input, it performs clustering, and then outputs a result in several formats that we'll be discussing in a little bit. It has different parameters that you need to pay attention to. The linkage parameter determines the definition for measuring. What it's going to be measuring is the intercluster difference. So if you set this to single, it's going to set the definition as the distance between the two nearest points in two clusters. If you set it to complete, it's going to define the cluster distance as that being between the two furthest points. The Ward methods are variations of Ward's minimum variance method. Then we can talk about the distance parameter, which specifies the distance metrics to use as far as measuring distance between your data points. The options for this are going to be Euclidean, Manhattan, Maximum, and Canberra. And then we have our clustered expected count, and it's going to set the cutoff height to the point and the one that results in a given number of clusters. There's the do transpose, which you need to add as true. And this allows for you to transpose the input matrix, such as to set whether clustered objects are going to be represented by rows or columns in the input table. And we can use this clustering to cluster objects or features. Let's do a little bit more examples with the hierarchical clustering pipeline. And let's go over the steps again, since there's a couple of them. Remember that the hierarchical clustering algorithm is a process by which a computer uses the input file where the columns are objects and the rows are going to be features. It's going to assign each object to a separate cluster or group, and then it's going to evaluate the all pairwise distances between those clusters using your chosen distance metric. There's going to be a distance matrix that's going to be constructed using the distance values. And we're going to find a pair of clusters with the shortest distance. Then all of these are going to be merged into a single cluster. And we're going to repeat from step 2 until we end up with a single cluster for all the objects. Our output for this is going to be a cluster dendrogram. And what I can say about this is that this dendrogram is going to look different based on the parameters that you choose, as well as the data that you start with. So for instance, this may not be what you get from the cell line data. This might be from the PDX data. So it just depends on what type of information that you put in is going to be determining which type of output you're going to get. There's another type of clustering that you're going to be learning about called k-means clustering. And so we can discuss the differences between those different methods. When we talk about the k-means pipeline that we input, here we're going to be inputting a table of expression in a .txt file format in. And after we run the algorithm, we're going to be able to observe 3 different things. The first of which is that k-means clustering doesn't do a great job of clustering. But next we'll find that basal is going to be split into 2 clusters, with cloud and low in one, and then normal-like in the other. And lastly, you're going to find that luminal A is split into 2 clusters. And so our output for this pipeline is going to be a .txt file as well, and it's going to include a cluster ID that each sample or object is assigned to. So you might be asking, well, what's the difference between this and the k-means? And the difference is that with the H-class pipeline, it has an output such as a dendrogram, which gives you a structure of your data. And the k-means is just going to give you the number for each cluster in each sample. So that would be the difference. But this is a good example of how different levels or heights might have different numbers or clusters or even subclusters. And so as we go to the top, there are going to be less clusters and the separation is going to be stronger. A good way to test any type of clustering model is to actually compare phenotypical data with the unsupervised classifications that you create. And then know that clustering is also widely used in data visualization. And it's used in order to organize the patterns into what are called heat maps so that you can visually see groups of samples and genes. Again here, very low would be red, and then going all the way towards high, you can see that the color is going to be changing to green. Large RNA-seq data collections provide us with the opportunity to discover signals in gene expression that aren't necessarily apparent with smaller sample sizes, such as prognostic indicators or predictive factors. And particularly, these are for subsets of patients. However, discerning the signal of each large data set frequently relies on the application of machine learning algorithms that are used in order to identify relationships in high-dimensional data, or to compute with, or to cope with computational complexity. These approaches often construct a model that capture relevant features of a data set. And the model can be used to make predictions about such data, such as how well a patient will respond to a particular treatment, or whether their cancer is likely to recur. So therefore, the model that you use is constructed using a large and diverse data set and is then going to be applied to incoming cases to make predictions about them. So let's go a little bit further and talk about different methods of classification, as well as how feature selection works. So this is going to be the third section of Transcriptomics 3. And so as you can see today, we've kind of covered quite a bit of material. Remember that classification is also called supervised machine learning. And this supervised machine learning is going to take in data that's been labeled. Typically, this is going to be done by people. And in projects or biomedical projects, this could be clinical data or some other type of phenotypic information that you get. The labeled data is going to be used to train a model, and the model is going to be tested on what's called a test data set. This data set will include examples of what's in the training data, but it might also contain additional variations. And the methods vary, as do the principles that are used to build the model. But for now, we can refer to them as a black box. This method will essentially learn from the differences between various classes that are present in your training data set. And once this learning is established, you have a model or a template that can be applied to new data sets. So new data will have to be assigned with a class that's present in the training data set. And if something doesn't fit in the model, such as the stars on the bottom right of the screen, your model accuracy will be greatly reduced. The output of your classification algorithm is going to include predicted classes for each object or sample that's found in your data. There's going to be other algorithms that you're going to be using, such as random forest, which evaluate how accurate and stable your predictions are. Sorry, what we have a question says, you don't understand the training data set. Yeah, so you're gonna have a training data set. We're gonna talk a little bit about that here in just a second. And we're gonna use an analogy of cats and dogs. So give me one moment and we'll be talking about the difference between a training set and a test set. But I was saying earlier, some algorithms such as random forest will evaluate how accurate and stable predictions are. And it evaluates which features, if you take them out, are going to be most significant for the accuracy of prediction. So let's go ahead and answer your question, Zinab, about training sets and test sets. And let's illustrate this simply. And what we can do is we can look at the classification of images. And we do this by training a model, and we can show it images of cats and dogs. And then we can test our prediction on. But instead of cats and dogs, we have gene expression. So to train the model, we have to label our data using phenotypic information, such as cancer stage, subtype, or risk factor. So our trained data has labels, and our test data has no labels. In the training data, we'll have a special row that we're going to call class, and it will contain labels for each sample. Genes in machine learning language are going to be called features, and our samples are going to be called objects. So the rows are going to be class, and those are going to contain labels for each sample. And then genes in machine learning language are going to be called features, and our samples are going to be called objects. The test data set will not have a class row. And so a test data set is only going to contain your genes, your sample names, and the label and the levels of expression. Another algorithm we're going to be using that's similar to this is called binary decision trees. And their output looks very similar to hierarchical clustering. Each one of these trees is going to be created by thresholds and rules. What binary means is that the branch can either be yes or no. And if the expression of a gene is higher than X, then the classes will separate. The algorithm is going to continue until it effectively separates all the samples into some group. If we use less genes in our training data set, it will give us different results because the data set can be dealt with differently. Additionally, binary decision trees are initiated in random start, so the genes selected in this procedure can change from time to time. And so here we can see cell line data that's separated into 3 groups. And those groups are going to be luminal, basal, and cloud and low with 2 genes. And then there's another procedure we can use called random forest, which is many decision trees. Random forest uses multiple instances of decision trees that are applied to our data in portions one at a time. After the analysis, the tree predictions are going to be analyzed and majority voting is accepted. The algorithm is useful for more complex patterns where smoother borders between classes are needed. And it also gives us an output of most significant features for classification. A natural extension of decision trees is this random forest algorithm. This is a technique that automates decision trees using a process of random initiation. The more a feature, or in our case, gene appears in these decision trees, the better it's going to perform. And this is an important point because random forest can help us to identify those features that are most useful for classification accuracy, as well as finding impurities. One technique that we can talk about that also has a pipeline associated is linear discriminant analysis, which is a generalization of Fisher's linear discriminant. This is a method that's used in statistics, pattern recognition, as well as machine learning. And it's used to find a linear combination of features that characterizes or separates 2 or more classes of objects or events. The resulting combination may be used as a classifier or for dimensionality reduction for later types of classification. LDA is closely related to another technique called analysis of variance, as well as regression analysis, which are also attempts to express one dependent variable as a linear combination of other features and measurements. Stepwise LDA, or SWLDA, is what makes it possible to automatically select those features in your data that are most useful or more relevant for the problem that you're working on. This process is also called feature selection. And then although both methods seek to reduce the number of attributes in the data set, dimensionality reduction does so by creating new combinations of attributes. How this contrasts is that feature selection methods include as well as exclude attributes that are present in the data without actually changing them. So here you can see for SWLDA pipeline, our input is going to be a train and test set. And these are going to be a gene expression table. And then we are going to get a plot from this. And that changing our Nouveau parameter is going to have different effects. So you can see here that we've changed it from 0.01 to 0.0005 to 0.00005. Remember that these inputs are going to be the same. And then for our case, we need to set this to the latter, which is going to be this bottom value here. In the results of our SWLDA pipeline, we're going to find that it's going to select only 9 genes. But what they will do is provide perfect classification for the training data set. SVM stands for Support Vector Machine. And this is an efficient classifier that can deal with complex separation between classes. And it's also very efficient. However, the SVM pipeline can have trouble working with too many features. The input for this pipeline is going to be test and train data, and the output is going to be predicted classes with accurate results for predictions. Let's talk a little bit about the applications of classification, which are methods that are commonly used in a variety of biomedical research, as well as clinical projects, where large data sets are beginning to become more and more available. These are going to include patient stratification, disease classification and diagnostics, as well as identification of potential responders to therapies. For the pharmaceutical and biotech industries, this could be biomarker discovery, as well as detection of toxicity at the in vitro stage. One of the other really important parts is going to be drug repurposing and repositioning. So that's all I have for today. I actually have 2 more slides. The first of which is that we're going to be talking about patient cohorts in TCGA on April 2nd. Same time, same place. And then the last thing that I wanted to talk about was our program for bioinformatics for infectious diseases. I'm going to post the link in the chat here. Some of you had the opportunity to come in and talk with us for the coronavirus webinar that we had on Friday, and I really appreciate you taking the time to join. I'd like to send you the program page for this, and hopefully you'll consider joining us for this session. We're not just going to be talking about coronavirus in this. We talk about Ebola and tuberculosis as well. We also talk about influenza, and we're going to be learning how to use bioinformatics to discuss some of these things and, kind of learn more. And we're not able to do this without you. So I hope that you'll take the time to come out and join us for this. One of the questions that we have, and I'm hoping that Mohit can help us if he's here, says if we could explain in the project Modeling Precision Medicine how to establish a relationship between classified genes and drug efficacy. So I think a lot of that has to do with, for instance, if the drug's working, if those are actually changing some of the...
Hi, Joe.
Hi.
Sorry, there's a lot of noise on my end, and so yeah, about this project, it would be better if I can go back. I have done this project, but I can go back and give Mikhail the answer he would be looking for. But I mean, for what I have understood is to first we are trying to classify genes and classifying them into several groups, and then using that data to reach to a certain point where we have a smaller number of genes, so that we can pinpoint to those which are those genes which helped us classify the into two subgroups, for example. So that's one of the objectives of the study. And drug efficacy is one of the feature or parameter which we see or compare. So when we are using machine learning, then we are using several features. So some of the features is like the gene expression data which we are looking at. So we are looking at transcriptomics data, and similarly it is another parameter to classify using the drug efficacy of how a drug has responded to a certain sample.
Great. Thank you, Mohit, for helping them with that. And by the way, if you have questions between now and the next session, try to post them in the forum, and I'll do my best to create some content around that information. But as I said before, remember that next time we're going to be talking about TCGA and our patient cohorts on April 2nd. And if you have anything, just let me know. And then I look forward to seeing you then.
Thank you, Joe.